you. Thank you for being here. Thank you for the opportunity. So as Gabriel said, the title of my presentation is quite long, so don't be scared. What we want to do basically is to use diffusion MRI to estimate biological properties of tissues. So that's it. This is very simple. And to do that, we will use a type of model which is called compartmentalized model. So first, I will motivate the work and why I'm doing this thesis. Then I will explain to you what is microstructure imaging and what is this approach. Then I will explain to you the methods I've been developing so far, which is called SEXI, which is a model for cellular permeable tissues, and the tools that we use to generate diffusion MRI signal, so which are Monte Carlo simulations. After that, I will show you the main results of this method, and I will show you how this method performs compared to other models in the field. So just to remind you, what is diffusion MRI? So diffusion MRI is an imaging technique that is sensitive to water displacement within the tissues. So on the left, you have an example of PGAC sequence, which is a gradient sequence that we use to acquire images. So what's important, it's the diffusion time that you have here, the big delta, that's separated in two gradients. So these gradients will encode and decode the position of water molecules inside the tissues. And what we measure is how far water molecules diffuse during the diffusion time. So how far they move from their initial position. Based on the sequence and the signals generated, we calculated some metrics. And one of them is the apparent diffusion coefficients that you have here, which quantify how much water moves during this diffusion time. Second part of the motivation is really cancer detection. So it's the application that we target in this project. So why do we, at the moment, cancer is detected by a biopsy, which is an invasive process where the doctor takes a sample of the tumor in the patient, and then the sample is analyzed in a lab. The idea of our project is it possible to do a virtual biopsy, which means is it possible to use diffusion MRI to detect the tumor without the need of taking a sample in the patient, which is non-invasive and very fast. So to do that, we will use microstructure imaging. So what is microstructure imaging? I will explain to you now. So we start from the tissue. In our case, this kind of tissue, let's say. This tissue, we put it in the scanner and we acquire the diffusion MRI signal. So nothing new now. But from an engineering point of view, this tissue can be seen as a mixture of biological entities that you have here on the right. These biological entities have different shapes, different size, different biological properties, and each of them has a different response to the gradient sequence. So for each of them, we can generate diffusion MRI signals. And the goal of microstructure imaging is to find the best combination of this compartment to recreate the full signal that we acquire in the tissues. So this is basically an optimization problem. And with the output of this model is really the contribution of each of these compartments to the full signal. So in the end, we can, well, so in the end, we have really the contribution of each of these compartments to the full signal, which give us the proportion of each type of cells, of each type of tissue in the sample. So this is the idea of microstructure imaging. So as you can imagine, this model are very dependent on the tissue that you have and the tissue that you want to image. In our case, we want to image cellular permeable tissue, so tumors, which are made as you have here on the left, of mainly spherical cells. So here on the left, on this part, you have an healthy lymph node, and on the right, you have a lymph node invaded by cancer, and you can see there's a huge difference between the two. And our goal is to discriminate these two tissues. So from my perspective, this lymph node is a sum of two compartments, which are an intracellular compartment and an extracellular one. The intracellular compartment is really spheres, and the extracellular compartment is water that moves around these spheres in the tissues. So here you have the equations of the signals, which are just for just to be complete, but they are not very interested in this case. What interests us more is really the parameters of this model. So you have 5 of them, which are first the intracellular and the extracellular diffusion coefficients. So for each compartment, you have a diffusion coefficient that appears in the equations here. Then you have the cell size, so really the cell radius. We have one cell size. At the moment, we have one cell size for the whole samples, but obviously you can see that you have different, in reality, you have different one here, of course. Then maybe the most important parameter of this model, if you compare it to what is existing in the field now, is really the permeability. So the membrane permeability quantify how fast water crosses the membrane of the cells. And finally, we have the signal fractions, which is the contribution of each of these compartment to the full signal. So what are the assumptions of this model? Obviously, we made the assumption that the intracellular compartment is made of spherical cells, which is not the case in reality. As you can imagine, the cells are not perfectly spherical. And from a diffusion point of view, we have 2 assumptions. First one is that we have slow exchange between the membrane, which means that the membrane are permeable, but not that much, let's say, which means that water will have time to move inside the cells before crossing the membrane. And the other assumption is that the compartments are well mixed, which means in this case that water molecule will let's say cross the entire cells before going out of the cells. Now that we have the model, we need the signals. And as I mentioned earlier, we don't use real signals at the moment. We use Monte Carlo simulations. So the goal here is really to reproduce and to simulate real acquisition, but with a computer. So for that, we need numerical substrates, which mimic really the tissue, but in the computer. So we started with white matter. So on the left, you have example of axons. And on the right, you have examples also of axons, but 1 year later. And you can see that this sample is much more complicated, much more realistic than the previous one. You have crossing, you have bending. It's keep improving from the previous version. But in our case, we are not interested in white matter, but in cellular permeable tissues. And so this is why at the beginning of my PhD, I implemented the same thing for cells. So these are the kind of substrate that I'm using at the moment. So you have an example on the left with a packing of spheres, basically, and the distribution of radii. But what is maybe most interesting for you is really how the simulation works. So you have a small animation here to show you. So we start from the substrate here on the left. This is the initial position of the particles for the simulations. And then for the diffusion time, we generate randomly the trajectories of the particles in the substrate. So here you have a sample of particles just to show you that some remains more in the cells than the others, for example, here. So now we have the model, we have the signals, then we need the experiments. to generate the results. So we start from the substrate. In the results I will show you, we use a substrate with 4 different radii, from 2 to 5 micrometers, and 4 different permeability, from 0 to 50 micrometers per second. In tumors, in literature, it's around 10, mostly around 10, just for you to have an idea of the tumor permeability. Then we did a gradient sequence. In our case, we use 4 diffusion times and p-value, 6 p-values up to 7 here. So these are a bit more technical, but not that interesting now. And we will compare our model with to Verdict, which is a model that was developed for tumors a few years ago, and that has some similarities and differences with our model, which are that they are both designed for tumors and they both model the intracellular compartment with cells. So as you can see, the intracellular compartment and the extracellular compartment are the same. The differences come from the third compartment, because as you can see, you have an additional one here, the vascular compartment, that models blood vessels in the tumors. And this model assumes impermeable membrane, which means that you have no exchange between the compartments. Now we are moving to the results, so to the parameter estimates. Before I show you the full results, I will explain a figure because they might be a bit complicated at the beginning. So for each model estimate, so for each parameter, I will show you this type of figure. The first plot on the top is really the output of the model, so it's the model estimates. On the lower plot, you have the mean absolute error, so really the error between the estimate and the ground truth value of the substrates. On the x-axis, you have the exchange time. So the exchange time, you have the expression here, is proportional to the radius of the cells and to the permeability. So if you have substrate with very high permeability, then the exchange time is very fast, and so you will be in this area. If you have impermeable cells, then the exchange time is infinite, and you will be on the left side of the plot. You have the same phenomena if you have big cells. So if you have big cells, the exchange time is larger, and the other way around. So if we come back to the plot, and you have different points on this plot. The color represents the cell size, and the symbols represent the permeability of the experiments. For each combination of color and symbols, you have one cell size, one permeability, and for each combination, you have 2 points on the plot. One is the model estimates, for example, this one, and then you have the same symbol with the black center, which represents the ground truth values. So basically, you have 2 times 16 points on the top, and the corresponding point on the bottom is the difference between these 2 points. So very simple in the end. So now if I move to really the model estimates and the results for noiseless data, I will show you the estimates for Verdict and for SEXY. So if we start with the cell size, what is interesting in this plot is that in the case of very permeable substrates, you can see that Verdict, so the plot on the top, is not able to estimate the cell size. While in the case of SEXY, the estimations are more stable, even if the samples are permeable. But in the case of impermeable tissues, so really on the left here, the results are better with Verdict, so the error is smaller. Now, if we move to the intracellular volume fraction, so really the contribution of the intracellular signal, you can see that with Verdict, as the permeability increases, the intracellular volume fraction is underestimated. So, and it gets worse. So as you can see here, really you have a decrease of the intracellular volume fraction and an increase of the error. While with SEXI, the estimates are much more stable for range of permeability, and the results are quite accurate, except at very high permeability. So in the worst case scenario. If you look at the estimation of the extracellular diffusion coefficient, we have the same phenomenon. So SEXI is much better than verdict in this case as well. Unfortunately, in the case of the intracellular diffusion coefficient, none of the model is able to estimate its value. So here it's really not relevant at all. So as I mentioned, SEXI takes into account permeability. So we are able to estimate permeability in this case, which might be the most interesting in the end. So if I show you the estimate in the case of noiseless data, you see that the estimate of the permeability is accurate, except at very high permeability, which is coherent with the previous plots. And this shows that even if we add one parameter to the model, which is the permeability, this improves. This makes sense because it improves the estimates of the previous parameters, and we are able to estimate this parameter as well. So it's really a good idea, let's say, to do that. Now, just to be complete, I'll show you the result with noisy data. And as you can see, the results are similar, except that we have higher variance on the estimations, which makes sense as well. So to conclude this presentation, SEXI provides accurate estimates if we are at reasonable permeability. But as I mentioned, in the case of tumors, we are in the range of this value, so really for the squares, which means that we are in the safe area, let's say. And it provides more stable estimate than impermeable models. So for SEXI, I showed the results for SEXI for a verdict, but we compare it with other models and the results are very similar. Unfortunately, we have some drawbacks that are that in the case of impermeable substrates, verdict is better. So which makes sense as well, because this model was developed for impermeable tissue. At the moment, we estimate only one cell size for the whole substrate, and our goal might be to estimate a distribution of radii or something similar. And finally, SEXI is not good at estimating the intracellular diffusion coefficient, but this is a common drawback to all compartmentalized models. So in the future, we would like to evaluate the performance on real data. So we are on our way to do that and develop a model of cancerous tissue that is much more realistic. So for example, by including other compartments or estimating a distribution of radii, or we would like to go in this direction. So that's it. I will just show you the references for the recording. And I would like to thank you and all the collaborators that contributed to this work. So thank you very much.
Thank you very much, Rémy. Do we have questions in the audience? Yes.
Like how important is the spherical assumption? Have you tested what happens if in your simulation you make them a bit elliptical?
The problem in this case is to generate that kind of substrates, because from an algorithmic point of view, generate substrate with a very high density of packing is difficult.
Yeah.
So that's the main issues. Of course, if you have some ellipsoid, it should be fine until one point the ellipsoid are closer to axons, which are really cylinders. So we will have a transition, let's say, from spheres to cylinders. And it will also depend on the diffusion time that you take. Because if you have a long enough diffusion time, then from a signal point of view, your diffusion signal will be the same spheres and ellipsoids. So,
And the competing model also assumes spheres. So,
sorry?
The competing model
Mhm.
also assumes spheres.
Yeah, exactly.
And yeah, last, like, why is that diffusion coefficient harder to estimate than the other?
I think that because it's confined inside the cells, the diffusion time is so long that you are not sensitive to really water displacement at short scale. So really what you measure is an averaging of the diffusion inside the cell, which means that if you have fast or slow diffusion coefficient, the mean distance crossed by particles will be exactly the same, I think.
Very, very interesting. So thank you for that, because I'm not in the field, but what I'm wondering is what prior information can you add to your model from histopathology?
Mhm.
And how can you, if I understood right, and it's quite possible I didn't, you're trying to detect tumors. So why not make the problem not an optimization of estimating parameters of the tumor, but differentiating healthy from pathological tissue? But maybe that's there and I just didn't understand it.
No, exactly. So you're right. The goal is to discriminate tumors from healthy tissues.
Yeah.
And the next step in this work is to fix the parameters of the model, generate the signals instead of estimating it. So in your case, we know from tumors, we know the cell size, we know the permeability, but what we are interested in the end, it's the fraction of the signal. So the next step is really to fix permeability, cell size, and estimate the signal fraction only. So yeah, that's the next step. You're right.
Thanks.
Thank you. Is there other question? Maybe on the Zoom call, if you have question. I don't see you, but maybe you can unmute yourself and ask question if you do have questions.
Turn on your cameras, especially so we see who's there.
No question online.
Maybe I am one. Just on your model parameter. So again, a similar question. So are you basing your assumption about the size and everything based on tumor or based on healthy tissue? So because I guess that there will be some changes, right? Structural changes in the tumor. So I might, but yeah, maybe you can comment on it. Whether if your parameter of your model are based on some data, some values that comes from these two different tissues, you will have different models.
So at the moment, we based our values on healthy tissues. So it's really this on the left. So it's not visible here, but here it's full of small cells, small spherical cells. And what we expect from LC to tumors is to have a signal fraction that is very different from this compartment, which we expect the intracellular volume fraction to go from, I don't know, 0.6 to 0.2 in this case. And this is what we would like to detect. Maybe it's the first step toward discrimination, and then we can also try to estimate permeability. Maybe there is a huge change in permeability between these two as well.
But my question is the model is optimized for healthy tissue when you present data from a non-healthy tissue? Would it be, and maybe you see something different, right? But is there an error in the model, or is there something that you estimate, from a real thing?
At the moment, because we are really estimating parameters and not, so we don't have prior knowledge as in machine learning or deep learning. The model is not trained; it's really a parameter estimation. So I think if your assumption has met, which means that in our case the cells are not that big, we should be able to see a difference between these two models in the parameter estimation.
Yeah. So there's a range where the model should work.
And that's why it's also important when you do some acquisitions to have the right PGSE sequence, so the real gradient sequence. Because if you have larger cells, you need larger diffusion time, which in practice is a problem because all the noise and all the stuff.
Okay, thanks.
Thank you.
Maybe just one quick follow-up question. Do you think the sequence should be adapted to the type of tissue you want to image, like different type of cancer or different type of, now you explore for diffusion time and 5V value, I believe. Do you think this is something to consider when you will be just tackling a specific type of tissue?
Especially here, we made the assumptions that the samples are isotropic, so the same in all directions. But for example, if you have tumors that are not the same in all directions, then you need to also take that into account when you acquire the sequence. So yes, definitely.
Any quick last question online on the Zoom?
So you get the difference results in model-wise, like, but in using permeability in the diffusion MRI signal. But if you have to, could say an estimate of the relative difference of a signal with permeability and non-permeability, how much could it be? Like percentage of difference in signal generations?
You mean really from a signal?
At the signal level rather than a model level. Like 50%, 100% difference, or 5% difference. So on a scale something.
So you mean the intensity of the signal?
Change. Roughly.
Again, it depends on the diffusion time and the value, but it decreases quite fast. So I would say maybe it's like 10% or per permeability increase, but it's really dependent on your parameters. So you also, of course, when you look at the shape of the signal, we'll go from fully impermeable to free water diffusion. And so you will have all the range in between, depending on your permeability values.
Okay. Thanks.
All right. Let's thank the speaker again.